And so I could do it after class one day, but I can't do it like without warning because I have a kid to pick up. So you have to like tell me that you're coming, and then you can come talk to me. So, um, okay. So, seems like I'm forgetting to say something. You just have your penguin blanket on. She's just like done with the world. She's just gonna wear a blanket. Okay. You look darling and super cozy. I know. I love penguins. Um, do you guys want to hear something cool about the aquarium? Do you know this about the aquarium? You can feed the penguins. You totally can. I got it for my son for a present last year. It was his favorite Christmas present. You can go in and feed the penguins. He's 10 and he's amazing. No, that's my 16-year-old. He's my stepson. I've had him since he was 3, but he doesn't live with us all the time. So you hear me talk about the other one more often. So yeah, you make an appointment. They take a limited number of people, right? They take you in like groups of 4 to 6, and you go in with the trainers. You put on all the trainer gear so that they think you're trainers, and then you get some dead fish in your hand with gloves, right? And then you just wait for little penguins to waddle up to you, and they eat out of your hand. It's the best thing ever. Oh my gosh. And my son was like, the 10-year-old. Mm-hmm. Yeah. Yeah. It's awesome. Okay. So we are going probably finish skin because you guys are a couple of minutes ahead of the other class. So the last thing we were talking about is skin color. Talked about suntans and sunburns. We were about to talk about melanoma. I wanted to show you one more picture of her. See? That one's a good one, right? The last class didn't see it, so I had to pull it up anyway. I know, it's so symmetrical. I think that I read somewhere, but I can't remember if I made it up. I think I read somewhere that vitiligo is generally pretty symmetrical in the way that it hits. Can't remember. Do you ever think that you heard something and you might have just dreamed it? Right? Happens to me all the time. Yeah. Like, did I just make that up? I might have. Okay. So let's talk about the cancer that occurs in your melanocytes. I'm going to remind you where your melanocytes are, right here, right? They're in most of us, they stay pretty close to the basal surface. In people with darker skin, you also have melanocytes that move up into the stratum spinosum, which is the next layer that we go. But either way, they're down close to that layer. So what happens if you have uncontrolled mitosis of these melanocytes? Well, it forms the most aggressive of the 3 forms of skin cancer, which is called melanoma. Melanoma only accounts for 5% of skin cancer occurrences, but accounts for the vast majority of skin cancer deaths. And the reason melanoma is a big deal is because melanoma metastasizes very quickly. It goes from one location into other locations in your body. And as usual, your textbook represents, of course you would know that that ain't right, right? So that's what melanoma looks like in the later stages. But in the earlier stages, like this guy has melanoma on his thumb, and it just looks like a dark pigmented blob. It doesn't really look inflamed or anything, and sometimes they're really flat when they start out. So here's a bunch of different melanomas. There is a way, now don't ask me to diagnose your moles because you don't want to die, right? Okay, you go to a dermatologist if you got an issue. But how do you figure out whether you should go to the dermatologist? Well, unless you're hypochondriac, if you think you should, you probably should. But there's a way to figure out, they say look at your moles, because of course if you've got dark pigmentation, it means you have a lot of melanocytes in that location anyway. And assess A, B, C, D. A is it asymmetrical? Generally, melanomas are asymmetrical. B is, does it have irregular borders? Do you see the borders irregular? And C, does it have color variation within the mole? Do you see that it's got color variation, at least most of them? And then D is the diameter. The larger the diameter, the more likely it is to be a melanoma. So Melanomas, these are also melanomas. These are a little less distinctive looking melanomas, but these are also melanomas. Melanomas, like all cancers, like all skin cancers, are more likely in areas that are exposed to UV radiation. But you know cancer's a jerk, right? It can and will occur anywhere, even in areas that have never been exposed to UV radiation. It's just statistically more likely if it's exposed to UV radiation. I have a friend who got a melanoma, get this, on her labia. And I'm pretty sure she wasn't tanning that way. It's just like cancer's a jerk. It's just like, oh, how about that occurs there? So yeah, it is not exclusive to locations, just an increased likelihood in locations exposed to UV radiation. So how severe is melanoma? Melanoma is a big freaking deal. It metastasizes very aggressively and very rapidly. You sit over there usually. What are you doing over there? It metastasizes very aggressively and very rapidly. I have a friend. She's 20-ish years older than me, maybe, I think. When she was still in college, so I'm from Texas, and I knew her at the college I taught at last in Texas. And when she was in graduate school, she went to graduate school up in New York, and then she would come home to Texas, and her mom would go, hey, you know that spot that's right there? You need to get that checked out. And she would do what we do, right? Which is blah, 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 and then go back and do what she was doing anyway, go back to school. And then she came home again, and her mom says, no, I think it's like a little different than it was before. You really need to get that checked out. And she didn't. And the third time, her mom made an appointment for her before she got there and took her from the airport to the dermatologist, right? And she didn't leave the dermatologist. She went directly from the dermatologist to the hospital, and she was in the hospital for 3 weeks. They gave her 2 years to live. This is how much tissue she lost. She was in her 20s, by the way, when this occurred. This is not a cancer of elderly people, generally. They took tissue from here up to about the tips of her scapula and down to about the middle of her sternum. All of that tissue, she had skin grafts from her butt. She had radiation and chemotherapy, and they still gave her 2 years to live. She's a success story because it did end up living. She also is the person who wears big floppy hats and opera-length gloves everywhere because she doesn't want any more sun exposure. So melanoma metastasizes very quickly. If you have a family history of melanoma, then you assume that you have a predisposition to melanoma. This and all of the skin cancers are at really disproportionately high rates in Utah because we have less protection from UV radiation at this higher elevation. And then this is the whitest freaking population in the country, right? I was floored when I got here. So I come from Texas, right? And I am half Native American. And so my family's got some diversity. And there was just always like a bunch of shades of people. And then I got here and I was like, where are all the brown people? What happened? What is going on? And my husband, who is 6'6" and blonde and blue-eyed, goes, oh, this is weird. This is weird. This is weird. I mean, if just having dark hair makes you exotic, you probably need some more diversity. Okay, so yeah, you got increased risk of all of those in Utah compared to the population at large. Okay, so now we're going to move to the next layer, and the next layer is the stratum spinosum. And so here we are. Let's just look at here real quick. So where we were is the stratum basale, which although it's hard to distinguish right here, it's of course the area that's touching the basement membrane, right? That's just the layer that's touching the basement membrane. And then the next layer is between there, the top of there, until the cells start looking like they have this dark pigment in them. Do you see that? That is called the stratum spinosum, and here it is in the figure. And it's, ranges up to 8 to 10 cell layers thick because it's variable in thickness, right? And sometimes 2 or 3 right here, so it's highly variable in thickness. It's called the stratum spinosum because the cells, some of them start to look pointy or spiny. So do you see that cell right there? How it looks like kind of like it's got a point on it? The cells start to get squashed so that they look kind of pointy, so they call them spiny cells or prickle cells. So that's why it's called the stratum spinosum, the spiny layer. Not all of them look like that, but so what's happening in this layer is that of course, the keratinocytes started out here. So concept check question, are the cells in the stratum spinosum younger or older than the cells in the stratum basale? Yeah, they are older. Their cell division slows down, but it's not zero yet. Like, for instance, this guy right here is still undergoing mitosis. Do you see that? Okay, so that's the stratum spinosum. Those cells moved up from the stratum basale. Since they're still undergoing cell division, our last type of cancer occurs here. And the cancer that occurs in this layer is the skin cancer that occurs in that layer. So they represent it in your textbook this way. It's called squamous cell carcinoma. And again, that looks pretty bad, but in its early stages, it doesn't look very bad. And this is probably the one that I would be the most likely to ignore because it kind of looks like, oh, I don't know, psoriasis or a scab that you pulled off too soon. It's like kind of rough and scaly and red, but it just stays like that and it doesn't heal, right? So doesn't that look like somebody just yanked a scab off too soon? Yeah, so that is how squamous cell looks in the early stages. Of the 3 types of skin cancer, this one is intermediate in its predisposition to metastasis. Now, don't get me wrong, all 3 types can kill you, right? But this one is intermediate between basal cell, which doesn't metastasize very quickly, and melanoma, which does. This is intermediate in its likelihood of metastasis. That makes sense? Okay, so that is squamous cell carcinoma. Okay, and then the next layer up is where if you stain it properly, you can start to see the precursor to hyaline. I'm sorry, to keratin in it. And so these cells right here, do you see how they start to look grainy and dark pigmented? That's not melanin. They're using a special staining technique because melanin would be mostly down here, right? Okay, so that is the precursor to keratin, which is called keratohyalin. And this layer is called the stratum granulosum because it looks kind of grainy, doesn't it? Yeah, go ahead, Brett.
So on the other picture,
this one,
yeah, is the basal, is it the
basal stratum basale down here?
Yeah. Is it just one?
It's just one layer right next
right
to.
above the pink and white.
Yeah, exactly. It's right above the connective tissue. Yeah, I mean it's not as easy to see it, but like you can tell where it is because it's just the one that's at the bottom. Okay, so this is the stratum granulosum. What the cells are doing here is a boatload of protein synthesis, right? Because keratin is a waterproofing protein, right? So what you're seeing is a whole bunch of protein synthesis going on. And this is pretty much the last chance that you have to do it, right? Because after this, then all the organelles are going to start to die out. You won't have a nucleus for transcription. You won't have ribosomes for translation. So you are doing protein synthesis right here. And then pretty much after that, the cells are just going to die and all of the organelles are going to die off. And so what you're going to have just above there, above the stratum granulosum, is a clear layer, which is called the stratum lucidum. So do you see, it's actually really thick in this one, but it usually looks more like this. Okay. So look at the thick skin, not the thin skin. Just look at this side. So you see the stratum granulosum, and do you see the kind of clearish layer right before you start the dead cells? That's the stratum lucidum. Okay, lucid means clear, right? If you have a lucid argument, you have a clear argument. Okay, so you cannot label the stratum lucidum in thin skin. Because and people say, oh, it's not there. Well, it's probably there, but it's like one cell thick. And you couldn't really distinguish it from anything else. So you're never asked to label it in thin skin, right? So this is sort of the in-between before you are just a dried bag of keratin, just that one little layer when you're before that. So that's the stratum lucidum. And then this next layer on top varies in thickness really dramatically. This is the stratum corneum. Corn like unicorn, it means horn, okay? This is made of dead cell membrane filled bags of keratin. And keratin you encounter all over the animal kingdom. It makes hair, it makes nails, it makes claws, it makes scales, it makes feathers, and it makes the horns of, not antlers, but horns like a rhino horn is made of keratin. So depending on what arrangement, you can either make it really hard or pretty flexible or those kinds of things. So in this context, it's functioning primarily as a waterproofing protein, a layer on the surface. So this is, of course, thick skin, and this is thin skin, but it has a stratum corneum on top as well. That makes sense? Okay, so let's talk a little bit more about the stratum corneum. So it varies in thickness pretty dramatically, but remember how we said that when these cells were formed, they were formed all the way down here, and they were hooked together via desmosomes, right? So when they move up and then slough off via exfoliation, they don't slough off individually as cells. So you usually slough them off in groups of hundreds, which is why you can actually see them when they're sloughing off. I mean, you don't always see your skin, but you can, right? If you tried hard enough, you could see a group of skin cells sloughing off. So that's the stratum corneum. And then we will move into the dermis. So before we do that, let's go back and revisit this picture for a second. And so what I noticed with some of you guys on your tests is that you made assumptions about what I was going to ask instead of actually reading what I did ask. So let me give you a couple of examples of that. I have no idea what your assumptions are, so I can't be held responsible for them. I'm just held responsible for what I ask, right? So here's a couple of examples. What if I asked you the layers of the epidermis from superficial to deep? Where would you start? Stratum corneum, okay. But what if I asked you from deep to superficial? Stratum basale. Either one of those questions is completely fair in my mind, but sometimes people have an assumption of what I'm going to ask. So you need to make sure that you're actually reading the question. And do you think that I should give it to you if you give me all of the layers but backwards? No, I'm not going to, because I'm actually testing you on two things when I ask you that question. Do you know the layers, and do you know the orientation? Do you know what superficial and deep mean? Does that make sense to you? Or here's another one. What if I asked not the layers of the epidermis, but I asked the layers of the cutaneous membrane from superficial to deep? Epidermis, dermis, exactly, right? Or from deep to superficial, dermis, epidermis. But do you see that sometimes people will assume that I'm asking one when I'm asking the other? If I say name the layers of the epidermis from superficial to deep, and then they put epidermis, dermis, right? That makes sense? So you just have to get, I know you have an expectation, but I don't know what it is. So you have to read. So you have to read what I'm actually asking. So, and a lot of times I can tell that you actually knew, but I can't grade that way, right? I can't just like grade, I think you actually knew it. So, because part of what you're learning is basically how to show that you know things, right? Okay, so now we are going to move down to the dermis. And we're not going to do the dermis in a whole bunch of detail, but just let's look at it. Okay, so with the dermis, it's mostly dense irregular connective tissue, but there's some loose areolar tissue in there as well. Remember that the dermis is the thing that kind of gives when you pull right here. Well, actually, the subcutaneous layer below the dermis gives when you pull right here. And it has a lot of stuff in it. In your textbook, pretty consistently, nervous tissue is represented in this sort of yellow color, so you can see how highly innervated it is. I don't know why, but I find it kind of interesting that when I do this, I'm actually not feeling it at all in my epidermis until the very bottom layer with a few of those little scattered cells, and I'm not feeling this pressure. I don't feel that in my epidermis at all. It's all in the dermis. All of that is, most of your sensory receptors are down in the dermis. So you have sebaceous and sudoriferous glands. Remind me what the simple term for each of those is. Sebaceous is what? Oil. And sudoriferous is sweat. Good. And then you have hair follicles, right? Okay. And these hair follicles, you also have lymphatic vessels. I don't know how much you guys know about the lymphatic system. Lymphatic system is not drawn on here, but basically you can drain excess fluid from your tissues via the lymphatic system, and we will talk about it later this semester. And then you have these guys right here, which are called erector pili muscles. And the erector pili muscles are responsible for making your hair stand on end, which doesn't seem very important or relevant in humans because you're a pretty bald mammal compared to the rest of them. But if you think about other mammals, you can see that it is actually a significant. If you think about how making your hair stand on end functions for a polar bear. What does it do for a polar bear? What? Yeah, it actually, so it's like putting on a down coat, right? So what happens is your body is generating body heat, right? And if you can keep it in the airspace close to you, it actually keeps you warmer, right? It's like fluffing yourself up. So it can be used for warmth. Or when your dog encounters the dog in the neighborhood that he hates, the hair stands on end, but it's not so much about warmth there. What is it actually doing? What? Is make them look bigger, exactly. Because we really don't want to get in a fight. Your dog actually wants the other animal to just give up when he sees them, right? It's just like, you are too much of a badass. I am not even going to try, right? So we do the same thing. We have hair standing on end in response to cold, and we also have it in response to stress. It just doesn't impress anyone, right? It's just like, oh, really? Yeah, right? You're like an entire, like half a centimeter bigger than you were before. I think I can still take you. Okay, so that's your erector pili muscles. I think, oh, highly vascular. It's really vascular in the dermis because not only is it responsible for giving nourishment to the dermis, but also the epidermis by diffusion. And then a couple of other things. So let's talk about dermal and epidermal ridges and papillae. Okay, so if you look at the surface of the dermis to the epidermis, it's not a smooth association between the two of them. So there are ridges. In some places in the body, the ridges are so dramatic that the epidermis follows the ridges. That makes sense? And those manifest as fingerprints and footprints. So what is the evolutionary advantage to fingerprints? What do you think? Grip, exactly. So fingerprints versus no fingerprints, or footprints versus no footprints, is like hiking shoes versus church shoes. Which one are you going to land on your butt in? You know, right? It's the church shoes. That's how you're going to land on your butt, right? So this actually increases grip, okay? That is your dermal ridges or your fingerprints. Now, your fingerprints are individual. Mine are going to be more like my siblings than they would be like yours, but ours are relatively highly individualized. So let's talk about them just a little bit because there's some interesting little tidbits about them. So when I was in grad school, I worked at a DNA fingerprinting lab, and my job to put myself through grad school was to do DNA fingerprinting analysis for primarily paternity cases, but we also did rape cases and murder cases and those kinds of things. So you had to do identification via DNA fingerprinting. So if I had, let's just theoretically, if I had identical twins, one of whom was a murderer, the best thing about that is that you always have an alibi, and that via DNA fingerprinting, I couldn't tell the difference between the two of you. You are genetically identical unless we start looking at your mitochondrial DNA, which is pretty difficult to do. So you're genetically identical. However, your fingerprints are different. And your fingerprints are different because of something really cool that happens during fetal development. So monozygotic twins share their fetal environment. They don't have two separate sacs. They actually share a sac. Okay? And they have genetically identical fingerprints. The predisposition for them, the pattern is identical. But what happens is they develop differently based on how you touch the sides of the womb and other structures when you're developing. So if one of you guys had your hand or your foot smashed up against the side of the womb, or really the side of the amniotic sac, really, then it actually slightly changes the development in your fingerprints compared to the other one. So what I would predict, if these two would let us see their fingerprints, we're going to be able to talk them into it, is that they would be far more similar than different, but they would be distinguishable from one another. So that is one of the ways that you can actually distinguish them. So when your mom wants to figure out which one of you guys did it, you can tell her to fingerprint. Like, you need to dust for fingerprints in the kitchen, because I'm pretty sure that she ate it and I totally did not eat it. That is fingerprints and footprints. Tattoos. So let's talk about tattoos. If you want a tattoo to be worth the hundreds of dollars that a nice tattoo costs, you should get it where? It has to go into the where? I'm sorry? No, where do you need it to go in the skin? Into the dermis, right? Because if you had a tattoo that only was in the epidermis, how long would it last max? 30 days, exactly. So that is, for instance, like a henna tattoo, which isn't really a tattoo. It's just sort of staining the tissue. And they don't last very long, right? They don't even usually last 30 days. They usually last, like they say, oh, it's going to last 2 weeks. No, it doesn't. Some of the good ones really do. Go ahead.
So on henna tattoos, it's actually how long you keep it on there. I've had one that's been on my hand for about a month.
Really?
So the
Oh.
more you keep it on there, the more it sinks into your tissue.
So it goes all the way through the epidermis, and then it can last as long as the
It doesn't
epidermis?
go through the epidermis, but
Because it permeates the epidermis if you have it in there for a long time. So yeah, so she knows where to get good henna tattoos, and I do not. Okay, mine are just usually on the street in Mexico when our family is vacationing, right? Same place you get cornrows, right? Don't think I ever did that because it's ugly. You can picture it, right? It's not a good look for me. Okay, so if you look at it histologically, what does a tattoo actually look like? An actual tattoo, not a henna tattoo. So you need to get this into the dermis. So here is obviously thin skin, right? And what happens is you inject the pigment into the dermis, and then your immune system has a response to that pigment. Hopefully the response is not a massively aggressive response because we've worked on the dyes for a while, right? So what happens is your immune system sort of encapsulates and cordons off that dye to keep it stationary in that location. And then as long as it is a stable dye, it stays there, right? Okay, so it stays there, and certain colors last longer. The pigments are better than they used to be for tattooing. But if you get exposed to UV radiation enough to damage your dermis, then this starts to break down the dyes as well. So if you pay a lot of money for a tattoo, you need to protect it. They're expensive, and the pigments are not everlasting. They don't last forever, so you need to protect it. So what about if I want to get rid of it? So I had my sweetie's name tattooed on my arm, and then she decided to sleep with my best friend, right? Not that that ever happens. But so what happens? So how do you get rid of that thing? You can do laser. So what happens is the laser actually permeates through here. It a lot of times does tissue damage on its way through. And what it does is breaks up the dye and breaks up the encapsulated area so that the dye is now small enough to be carried off by white blood cells in your system. That's how laser tattoo, huh?
Where does it go when it's carried?
Just wherever white blood cells, just break it down and dump the trash into the liver, kidneys, skin, wherever, just as long as they're small enough. Go ahead.
so does that actually work the same as burning the tissue itself with the laser?
Well, it depends on what kind of laser, right? The specificity of the laser is determines how far it goes. But could you actually just use heat? You can use heat. That would actually destroy it as well, but only if you actually used heat enough to damage the dermis, right?
So the laser does not damage any of the tissue?
No, it does. Usually the laser actually does some tissue damage on its way down. They've gotten better over time, but a lot of times people don't have a tattoo anymore, and they do have a slight scar tissue formation where the tattoo was. And it's getting better over time. So basically you really need to think about tattoos, right? Okay. Make sure it's nothing to do with another human. Okay, because have you met humans? They generally suck, right? 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 Maybe your dog. Your dog is never going to do you wrong, right? I'm going to get Maddie's name tattooed right there. Golden retrievers are just loyal, right? She's going to love me forever. Okay, so, and then the last thing is the dermis. I know it's gross to think about, but it's worth talking about. The dermis is what you tan. You stretch and moisturize and stretch and moisturize. Keep the flexibility in it to form leather. And then the reason leather is good at what it's good at is because it is dense, irregular connective tissue, right? And it's strong in a bunch of different directions. And so although we generally tan the dermis from another mammal, you can use reptiles, right? Alligator boots. You can use the dermis from other organisms. And it has been used from humans before. Not generally very good stories, although some of them are kind of weird. So I'm going to tell you this weird story just because I thought it was so freaking cool when I actually encountered it. So you guys know that if you talk about any religion enough, it's going to sound really weird, right? Just keep telling the story, even if it's your story, right? And you're going to start saying something that sounds weird. So if so in all over Europe and in South and Central America, the bodies of the saints, this is in Catholicism, by the way, the bodies of the saints are considered holy. And all of the pieces of the bodies of the saints, they're considered relics. I don't know if you've heard of relics of the saints. So like St. Ignatius, if you like have his finger bone left, it's going to be in this beautiful case. You've got like the larynx left from somebody, and you have like just portions of the body left. And it's not unique to Catholicism, but this is the context in which I know it. And so those are really holy, and people come to pray at those sites and pray to those saints and all of that kind of stuff. Well, what if you wanted a Bible to be extra special holy? What would you cover it with? Do you think I'm joking? I am totally not joking. You make the covering of the Bible out of the skin of a saint. And then it's extra special, super duper holy, right? I'm telling you, don't even, just start talking about your own religion out loud and you'll start to hear the weirdness in all of it, right? So yeah, I can't remember the names of those. I was trying to remember the names of those books. So if one of you guys like super techie people, you have my permission to look it up right now. Find out the name of like a holy book covered, and sometimes it was prayer books, sometimes it was Bibles, holy book covered in saint's skin. Just find it out. We want to know, right? It's going to come up in a trivia question, and it's almost Halloween, right? You're going to need it. You're going to need it. I love knowing weird crap like that. Okay, so now that we are properly creeped out and think, remember I told you how much humans suck? Right? Okay, so now we can move to the hypodermis. And the hypodermis is also called the subcutaneous layer or the superficial fascia, and it's down here. Okay, and it is a combination of adipose tissue and what kind of tissue? Loose areolar connective tissue. How much adipose versus loose areolar has to do with how heavy you are, but also where it is on your body, because of course you deposit fat in certain places versus other places. That area is going to be really, really vascular. There's lots and lots of blood flow to it. It's deep to the dermis. It is the part that actually gives. So the dermis, let's go, did you find it out?
One says Destinies of the Soul.
Destinies of the Soul? I thought it was just a name of the book.
I don't know.
You didn't find it yet? Oh, you did. Arthropodermic. Let me read that. I just need this information. Anthropodermic bibliopedy is the practice of binding books in human skin. Yes. And so 17 books have been confirmed as having human skin bindings, and 9 were proven to not be of human origin. Anthropodermic, right? Anthropo means human, dermic means skin. Biblio, but I don't know what the suffix pegi means. Do you? I don't know. So the more you know, right? Okay, so the hypodermis, back to the hypodermis. About 50% of your body fat is stored there. Of course, it's highly variable, right? Depending on how heavy you are and where you're talking about. And it increases in thickness dramatically with weight gain, but here's the interesting thing. It increases in thickness dramatically differently under the presence of estrogen and testosterone. So let's tell the story of how you get a body shape of an adult. Because if you look at 2 little 7-year-olds, right? Not only are males and females not dramatically different in body size at 7, right? The most of that happens at puberty. But they're also not dramatically different in body shape at 7. So if you looked at 2 little 7-year-olds from 25 feet away, they don't look very different in their body shape. They're just like kid shaped, right? Just like a kid-shaped kid, okay? But then they hit puberty, right? And a lot of things change at puberty, but what I want to concentrate on here is body fat deposition. And so now I'm going to look at a 19-year-old and a 19-year-old, 2 different genders, right? And you can tell from far away by the body shape which one's male and female most of the time, right? And that is because under the presence of estrogen, it changes your body fat deposition from the little tadpole body fat deposition. You know how kids are like the only people who are cute when they have fat bellies, right? They're like adorable, little twig legs and little big old fat bellies. It's adorable. It's not that cute when you have it as an adult. When you have estrogen, here is how estrogen changes your body fat deposition. Estrogen makes you put less body fat on right here, so you tend to get a waist. and more body fat on right here, and more body fat in the breast. Okay, so you get this shape. Okay, and then in males, testosterone changes your body fat deposition differently than estrogen does. Males, when they hit puberty, tend to put less body fat on their legs, so they get little chicken legs for a while, right? And then if they have body fat, it tends to go up here, right? So from the belly up to the little man boobs that sometimes take a little while to stretch out into the body, all the way up to here, right? Sometimes body fat up here. And so the shape of males tends to be more like that, and the shape of females tends to be more like that, right? And that is the presence of your hormones. And of course, males also grow generally more. They get taller than females. So that's kind of interesting. There have been some studies that I find so fascinating. So I'm really interested in the evolution of human sexuality. I've studied a whole bunch of it. My background in grad school was evolutionary biology and molecular biology. So one of the things that they figured out that doesn't vary very much from culture to culture, because a lot of when we're thinking about how humans find other humans attractive, we think, oh, well, that's very cultural, what you find attractive. One of the things that seems to be very consistent from culture to culture is that heterosexual adult males have a preferred waist to hip ratio in women, okay? And they don't even know that they have a preferred waist to hip ratio. You just manipulate the photos. You saw the same photo of the same woman 25 photos ago, and you manipulate it so the waist to hip ratio is 0.7, and all of a sudden, you are fascinated with everything about her. You're like, oh my God, I bet she's funny too. Or listen to all of her stories. So it is the evolutionary trigger, it seems, in male for fertility. And I know you're not trying to make babies, but part of you is trying to make babies, right? Evolution's trying to make you make babies, right? Wow. So yeah, they manipulate photos. Now, it doesn't have to do with you being thin, because some people like thicker, right? And some people like thinner, but the ratio is what seems to be very consistent from culture to culture. Because, of course, you know, in some cultures, thin women are not considered attractive at all, right? And some cultures, thick women are not considered attractive at all. But what we seem to almost universally like is that shape, that waist to hip ratio. So you can go home and get your measuring tape out, right? No, I'm just kidding. But women don't care about that, just so you know. Women do not seem to be impacted by the waist to hip ratio in males at all. What women, if you can manipulate photos of men, there's all different ways that they manipulate photos, so that it looks like they have more testosterone, as indicated by their facial features, so you get a bit stronger jaw and a bit stronger brow, then women tend to find them more attractive. Okay, so we're basically, it's just an arms race between the genders, just so you know, right? We're just trying to manipulate each other. Okay, so that is estrogen, testosterone. I already talked about that. Okay, so in this picture, you can't see everything, but what you should be able to see is to tell the difference between the hypodermis, the dermis, and the epidermis, right? And then in the epidermis, you shouldn't be able to label all the layers, but you should be able to point to the stratum corneum and the stratum basale, right? So even a bad figure, you could tell all of those. Good. Okay, so let's see. Now we're going to talk about what happens when you damage your skin and how it heals. So the skin is also called the integument. This applies to other tissues as well, but integument, repair, and regeneration. So here's plan A. If I damage a cell, I replace it with the same cell, I lose no functionality, right? Doesn't that seem like what would be ideal? You damage something, you put it back, and you don't lose any functionality. That's not always possible. When it's possible, that's definitely what you would want to do, and that is called regeneration. So damage a cell, replace it with the same cell, don't even notice any difference later, okay? Now, when you can't do that, one of the reasons that you can't do that is because regeneration is relatively slow. And sometimes it's more important to plug the hole than what you plug it with, right? So I want you to imagine that I've got a big, nasty wound on my arm, right? So regeneration is ideal, and it has to start from the edge, right? Because you actually have to have living cells to go through mitosis, right? So you start from the edges and you start regeneration. And then your body's like, oh crap, I have a big freaking hole that's going to let a bunch of pathogens in the middle. And so what is faster and is plan B is scar tissue formation. and that's called fibrosis. So it's basically like spackling the hole, right? You got to like fill the hole with something because you're running into threats to your immune system at that point. So the process of fibrosis is scar tissue formation. So let's talk about how that occurs. First, let me introduce you to this word that you may or may not have met. Parenchyma is not a specific meaning. What it means is the uniquely functioning cells in the tissue. So stick with me for a second. Parenchyma in liver is the hepatocytes that are unique to the liver. Parenchyma in like skin might be the keratinocytes, but it's not the stuff that exists in all tissues. Like for instance, you might have fibroblasts in all tissues, right? But you have lung cells in the lung and pancreatic cells in the pancreas. So what happens here in fibrosis is you damaged parenchyma and you replaced it with connective tissue, right? So do you see why I call it spackling, right? Like at home, you had drywall, somebody punched a hole in the wall, right? And then you spackled over it, right? So it fills the hole, but it doesn't function exactly the same way. So now here's the process of fibrosis. The process of fibrosis is basically, well, first you better stop bleeding. Wow, it's interesting out there. First you better stop bleeding, and so you're going to have to do some clotting. And what will help with stopping the bleeding is, although it's counterintuitive, you need to bring blood vessels into the area that bring platelets and clot. Then you actually have more capillary growth, and as you increase blood flow to the area, you can also bring in white blood cells. White blood cells are important for protecting you, but they're also important because they clean up the waste, right? So all of the dead and damaged tissues is scooped out by the white blood cells. Okay, and then the next important part is remember that you always have hanging out by your blood vessels, you always have mesenchymal cells. Remember, those just are always around your blood vessels. These mesenchymal cells are now going to differentiate into fibroblasts. And then the fibroblasts are going to do what fibroblasts do. What do? They multiply and they make fibers. And now you get to play God. What kind of fibers are you going to use to repair this hole? What do you think you're going to use? Huh? Just the fibers. Collagen. You're going to use collagen. And so they start to form collagen from the bottom up. And so you're knitting this wound from the bottom up, right? Okay. And it's healing from the, isn't that how wounds heal, right? They heal from the bottom up. It's knitting it together, but what it's not doing is remaking all the cells that were there before. So if I had a hair follicle, I can lose it. If I had a sweat gland, it might be gone, but I am knitting it together. So I'm spackling it up from the bottom to the top. And by the time I am almost done, I still have a lot of blood flow into the area just because I'm still working on it. And then I have a whole bunch of collagen fibers, and I have that pink tissue that's almost healed but isn't. You know what I mean? It's like the tissue that would be under the scab. That's called granulation tissue. That tissue is not done yet, and it's really easy to re-injure. But then if you give it a few weeks, you are done, and you've made a whole bunch of collagen, and then the capillaries atrophy. So at that point, the scar, which is what it is at this point, generally is paler than the tissue around it. The capillaries leave, right? That makes sense? And it's lost some functionality. So scars don't usually tan the way the other tissue does. They don't usually sweat. They don't usually grow, produce oil. And they usually lose a lot of elasticity in the process, right? They're usually not as elastic as the tissue around them. Go ahead.
If a nerve ending is lost, will that regrow or will it?
Well, when we get to the nervous tissue, I can say that it is not likely to regrow. Scars are usually slightly less sensitive than the tissues around them once they're completely healed. During the healing process, they're really sensitive because you've got exposed nerve endings. So a lot of times you'll lose nervous system sensation. But nerve healing is a whole different story, and we will talk about it in the nervous system. Okay, so what you end up with is a pale, usually pretty avascular scar. It doesn't have much blood in it. Now, scar tissue is a big deal because the last step of scar tissue formation is that the collagen fibers sort of pull the edges together. And that's all well and good if it's on the surface, but if it's at a joint, that can actually reduce mobility. Or what if it's in a tube, like fallopian tubes? If you have scar tissue formation in a fallopian tube, as it heals, it actually kinks the tube. So that last stage of scar tissue formation, you felt that, right? When your scar is just about done, you can especially feel it with like a road rash. If you fall off your bike and get a road rash in the last step, it's like tightens, right? That's okay on the surface, but if it happens at a joint, it can reduce mobility, and if it happens internally, it can kink and change the shape of tubes. So, last question is, why would you ever do this? Why would you ever do fibrosis and not regeneration? Well, first answer was the size, right? Because regeneration is faster than fibrosis. But then the second answer is some tissues can't do regeneration. Some tissues have lost their capacity for mitosis. And so you don't leave holes. You have to fill it with something, and you are generally going to fill it with scar tissue. So here are the 3 categories of cells in your body. And this is not a specific name for a cell. It's just their predisposition to mitosis. The first category of cells are called labile cells, and those are undergoing mitosis all the time anyway. And so if you damage them, they're very likely to go through regeneration unless it's a huge wound. So great example, the skin on the inside of your mouth. How many times have you bitten and chewed on that, and does it have scar tissue? No, it doesn't generally. I mean, if you damage the entire side of your face, I bet it would, right? But it actually has a really good capacity for regeneration. Your skin also does, so you've had far fewer scars than you have had injuries, right? And the bone marrow, that has to be labile cells, otherwise I could never donate any to anybody. Has to regenerate, right? Okay, and then stable cells is the next one. They are not always going through regeneration, but they remember how. And this is like a lot of your glandular tissue, your liver can, your pancreas, those kinds of and endocrine cells, endocrine glands. And then the last ones are the ones that you never want to damage, and these are called permanent cells. And permanent cells have 0 capacity for cell division in adulthood. And these are the tissues that you really want to protect. Like, for instance, cardiac muscle. Cardiac muscle is amitotic in adulthood. So if I have a heart attack, right, my cardiac muscle cells die off, right? And I don't replace them with cardiac muscle cells. I replace them with scar tissue, which means what about the strength of my heart? No, it's actually weaker than it used to be, right? And you're more likely to have a second heart attack. That's just really, that's some piss poor planning right there, don't you think? I mean, what is the function of this institution? What is it? Okay, so, and then your brain, you never, right? Your nervous tissue, most of your neurons are amitotic. They used to say all of them, but there are a few of them apparently that can be motivated for regeneration, but most of them are amitotic. So let's talk about a stroke just with our last minute. So if I have a stroke in the right side of my brain, you may or may not know that that would affect left side function. And a stroke kills off the tissue, and I don't get the tissue back. So I fill the hole with the brain's equivalent of scar tissue, okay? And I have lost that functionality there. Now, does that mean with the brain that I can never regain the function? With the brain you can, because the brain has something called neuroplasticity, and given some time you might be able to pass off the functionality to another part of the brain, but you're never getting the tissue back, right? So if you have a stroke, you have a scar there on the brain. I mean, it doesn't look quite the same in the brain tissue. Okay, so we will stop there. We'll do this next time, just skin throughout life and then burns.
Why does it scar?